Good evening, everyone. My name is Himanshi Singh, and I'm a PMRF PhD candidate in IIT Kanpur at VSB department. I welcome you all for the week 8 discussion, previous year discussion, specifically for the course Animal Physiology, which is taken by Professor Manik Das, who is also from IIT Kanpur itself. So I hope that you guys must have went through the week 8 lectures. There were 5 lectures and we are still continuing for neural system. As you might have noticed, like last 2 week was also dedicated to only neural system. And I guess in the last class I have discussed why we are so much interested and intrigued, and we want more detailed oriented information specifically for our nervous system or neural system, right? So in this week, specifically for week 8, we discussed the extended nervous system, meaning we went for systems instead of looking at the more keenly into the junctions of neuronal system, such as types of neurons, how they connect with each other, how they share information, which we were seeing in the last 2 weeks, right? So we went to for olfactory system, which is our smelling system, right? We also went for auditory system, which is our hearing system. And in the coming week session, I guess we're gonna also talk about our vision system, that is our eyes, and how it works. So I hope by this time you guys have pretty good understanding about a neuronal system or nervous system. At least some basic information you can now see what is an excitatory neuron or what is a inhibitory neuron, right? What are their functions, how they contact with each other, how they make connections with each other, how precisely the junctions are regulated, right? How precisely the information is being carried out. The information is transferred in the form of chemicals as well as electrical impulses, right? So different types of neurons, myelinated, non-myelinated, right? Central nervous system, peripheral nervous system, cells, neurons, which are associated with these two systems. So even if you do not, you are finding it difficult to remember everything, I will suggest you to just try to have this mind map of whole of the nervous system, that what are motor neurons, what are sensory neurons, what is a skeletal muscle. The skeletal muscle will be innervated with neurons, right? So having a mind map will allow you to have a clear image of whenever you are studying or reading. It will help you understand at what place of that mind map you are reading, right? Because it's a huge system to understand. So I hope that you guys have went through the week 8 sessions, and you whenever you have any doubt, just you can either drop me a mail on the website which I have mentioned at the bottom of the current slide, or you can drop a comment on the YouTube whenever I'm gonna upload the video. And as usual, I'll recommend you to always keep on track with the current week lectures. With this small step, you won't be having a lot of burden upon you if you have signed for the certification course, if you have registered for the certification course, that you have to mug up or learn, because this is a huge course, right? It's a 12-week course and we are halfway through it. So it's a lot of information to pack in within like 2 to 3 days. So I'll recommend you to watch the sessions regularly, attend live sessions, ask question, discuss. This is the best way to learn and understand the subject. It is a very intriguing subject because we are knowing about ourselves, how we see, how we like perceive ourselves, how we think, and all those stuff related to oneself, right? So in a way, it's an interesting course, and I hope that you guys are enjoying this course. With this, I guess we can start um, today's session. And as usual, I'll start with a little bit of information, which will be an extension of what you have already covered in the current week, that is week 8 for today's session. And this discussion is not meant to like mug up and learn and all those things. It is not meant for this. This open discussion is for you guys to understand and talk and think freely about a topic and how to connect the information which you have with the knowledge you can create with this particular information. Okay. So moving on with this. So as I mentioned that in the current week, the extension of the nervous system, which is a whole like connection of neurons, muscles, receptors, right? And small molecules. It's a system. So I would like to discuss particularly about the olfactory system today. In the lectures, sir has discussed about the olfactory system as well as for the auditory system, but I want to take this olfactory system from a different point of view, because in the lecture you have understood olfactory system from nervous system point of view, how neurons are connected, how we smell, how we sense smell, and all those stuff. But I want you guys to see the same system from different perspective. And what is that perspective? That perspective is from receptors point of view, right? And by receptors point of view, what I want to convey to you guys is that if you remember our discussion on biomembrane or a plasma membrane or a cell membrane, which we had, I guess, maybe last to last week, I do not remember exactly. You can go back and check that discussion. It is in the beginning of the session itself. So we have discussed about the importance of cell membrane. We have discussed about what cell membrane is comprised of. We also discussed what cell membrane, what are the different components of a cell membrane, right? A membrane is made up of lipids, correct? Lipid bilayer it was. And what are the things which are present in the membrane? Then we have seen certain proteins, right? And those were different types of protein. A protein which is only associated with the upper leaflet or one leaflet of a membrane, right? Those peripheral membrane protein. Also a protein which is associated only with the bottom layer or the inside layer of a lipid membrane. That was also a peripheral membrane protein, right? There were certain proteins which were completely transversing both the layers of a plasma membrane. We call them transmembrane proteins, right? And there were certain proteins which were associated with the lipid with another lipid head group, right? With the help of a lipid moiety, they were connected. They were called lipid anchored proteins, correct? So these are different types of proteins which are associated with the membrane. But even the proteins belong to different classes of protein, right? Depending upon their structure, their function, to whichever angle we want to see, we can understand different types of receptors. Okay. So today, what I want you guys to understand is that what is the need of a receptor at the end of the day? Why we need receptor? Correct. The need of the receptor is to establish a communication between the external environment and the internal environment. Correct? The external environment means from where we are receiving the signal. And here I'm talking about the cellular space, not the external space which you can see. So if inside your body there is a cell, it needs to, for example, produce some chemical compound, say insulin, say beta cells which produce insulin, right? So beta cells should have something in them which will tell them to produce insulin. Correct? So what is that something? That something will come from our gut, because when our gut is full, our system is saying, okay, we have enough glucose. Now stop using it, right? Keep it storing. Correct. Now we need to store it because there's a lot of glucose. We already have enough glucose in our blood, and if we will not store it, it will get into our blood, and it will lead to a disease called diabetes. Correct. So the signal will come from the outside to the beta cells. Now that signal which is coming, how the beta cell will detect it? There has to be something on the cell surface, right? Which will tell them that, okay, there is some signal coming. I don't know what that signal will do, but let's see what it will do. Correct? So if a signal comes to the beta cell and it binds to a receptor, that receptor will undergo certain changes, or it will start a cascade of reaction inside the cell, which will lead to the cell and it will signal the cell to make more insulin, or depending upon glucagon, like whatever the signal was received. Now this receiving site, this receiving part is done by the receptors, correct? And that is why I mentioned that receptors are the ones which will help us in converting the outside signal into a action. And here action is not action potential. Action here means anything which will lead to a biochemical change, and due to that molecule or ligand or what substrate or whatever you want to name it, it will lead to change in the cell biochemistry, leading to some changes or as a result, correct? So if we talk about receptors, there are different types of receptors. Receptors are membrane protein, and we have discussed broader characterization of membrane protein as peripheral membrane protein, lipid anchored membrane protein, transmembrane proteins, right? So among them, the receptors are the ones which will have a binding site for a ligand or a small molecule or a signal, say signal here, right? So depending on the type of ligand or the type of function which it is associated with, receptors can be categorized into multiple classes, okay? But a very generic class you can see here. This is based upon their function as well as their structure, okay? So if we look at the first type, that is the A type of receptor, it is the ion channel receptor. Now what ion channel receptor does is that it will transport or it will sense the ions. Now it can be a cationic receptor or an anionic receptor, okay? We are not going in deep detail about it, okay? Second is the 7-helix transmembrane receptor. What does 7-helix transmembrane receptor means? If you count, transmembrane means the protein will be crossing both the bilayers of the protein, both the bilayers of the membrane. And here 7-helix transmembrane receptor means that there are 7 helices. Helix is the secondary structure of a protein, okay? So there are 7 helices which are of what nature? They are of transmembranic nature. And this comprises as one single receptor. So what is the structure of this receptor? It is 7-helix transmembrane. So you can count. This is first helix. There's a turn. This is second helix. There's a turn. There's third helix. There's a turn. There's fourth helix. There's a turn. There is fifth helix. Then there is long domain. Then there is sixth helix. Then there's a turn. And then there is finally 7th helix, right? So this is a type of receptor which has 7 transmembrane helices. Then another type of receptor is called receptor with intrinsic enzymatic activity. So what does intrinsic enzymatic activity means? It means whenever a small molecule or a ligand or a substrate will bind to this particular type of receptor, this receptor in its cytosolic side will itself act as an enzyme, which means it will convert a substrate to a product. It can be any substrate or product, okay? We are not going in that much details. The next one is the enzyme associated receptor. So what does enzyme associated receptor means? It means whenever the ligand of this particular type of receptor comes and binds to its receptor binding site, there will be certain changes in the cytosolic side of this receptor, which will allow other small enzymatic type of proteins to get attached to this receptor and then perform its function. In the first one, the receptor itself was having the enzymatic activity. In the second one, the changes leads to the recruitment of enzymatic active moieties, which will do the work of converting substrate to product. Okay. Now, whether you have heard about it or not, but the 7-helix transmembrane receptor, this is a very different setup of receptor, oftenly called as GPCRs. So what does GPCR mean? GPCR means G protein coupled receptor. So in 7-helix transmembrane receptor, there is a class of protein which we are aware of, which has the 7 transmembrane helix structure, is called GPCR, right? Yeah, it is called GPCR. GPCR means G protein coupled receptor. Okay. So as the name suggests, that G protein coupled receptor, which has a structure of 7 transmembrane helix, has a G name of a protein as a type of protein which is called G here. Okay. So G protein, and this receptor is associated with this G protein. Okay. Now, why I am putting effort on GPCRs? Because the whole olfaction system, right? We smell something, we sense something. So in the lectures, you must have, if you have gone through this week's lecture, you must have seen that there were odorant molecules which Sir was talking about. Those odorant molecules were binding to a receptor, and then the neuronal signal was going to the olfactory part, olfactory sensing part of our brain, and then the whole information was relayed. So that receptor, it belongs to a class of GPCRs, which has a structure of 7 transmembrane helices. Okay, I hope that this much is clear with you guys. So GPCRs meaning G protein coupled receptor. And why I'm focusing on it? Because majority of your body senses, for example, your smell sense, your vision sense. In the next week, I guess we will see about the our vision system. Even that vision system, the receptor which is functioning at a molecule, at a sole point source level, that also belongs to a class of GPCR. So now you can understand the importance of GPCRs, right? That our smell, our vision, and other aspects also are connected with only one class of protein, which has a structure of 7 transmembrane helix, indicating that this 7 helix transmembrane receptor structure is important, right? And it's also a characteristic of GPCRs. Now, this G protein, which is coupled with this receptor, will have different functionality. So we are not going into deep signaling or like cycles of GPCRs, which it can stimulate when the G protein coupled receptor is stimulated with its ligand. But I want you guys to understand that the available drugs, right, which are in the market, and I'm not talking about any particular disease or any particular group of diseases. I'm not talking about that. I'm talking in general. Whatever the drugs which are available, around 30 to 50% of those drugs are targeted only against GPCRs. It's a huge number, right? We have a lot of drugs in the market which are FDA approved, sorry, which are FDA approved, correct? Most of those drugs work on GPCRs because GPCRs are so common in our body that if we are looking to treat any disease, chances are that it will be associated with a GPCR, right? So talking about the olfactory receptor or the olfactory system, so our olfactory receptors, also called smell receptor, are proteins capable of binding odor molecules that plays a central role in the sense of smell, correct? These receptors are common to arthropods, terrestrial vertebrates, fishes, and other animals. In terrestrial vertebrates, including humans, the receptors are located on olfactory receptor cells, which are present in a very large number, millions, and are clustered within a small area in the back of the nasal cavity, forming an olfactory epithelium. Each receptor cell has a single external process that extends to the surface of the epithelium and gives rise to a number of long cylinder extensions called cilia. These cilia are covered by the mucus of the nasal cavity, facilitating the detection of and response to odor molecules by olfactory receptors. Right? So we have millions of these GPCRs or olfactory receptors, and yet we are not the smartest one in using our olfactory system for survival, right? We use our olfaction for day-to-day work, but we do not use, or we are not completely dependent upon our olfactory system. Imagine about a dog. Dog has a really good sense of smell, right? So dogs have much more these olfactory receptors in comparison to us. Why? Because we mostly rely on our vision system. So our other system is not that much developed in comparison to other organisms. Correct? That is why when we say that olfactory system is one of the system through which animals survive, this statement is not true for us, because we depend mostly on our visual system for making judgments, for sensing a risk, or for just judging a situation, how we should react and how we should act. We majorly depend upon visual system and not on olfactory system. Correct? So to understand these GPCRs and what they look like, I have these small videos which will help us in understanding the importance of GPCRs as well as olfactory system. Okay, so let me quickly start this. Just give me a minute. If you can hear. I found this video very, very intriguing. So first we'll see how we are sensing the smell. Okay. I hope you guys can see this. Yeah.
It's the first sense you use when you're born. 1 out of every 50 of your genes is dedicated to it. It must be important, right? Okay, take a deep breath through your nose. It's your sense of smell, and it's breathtakingly powerful. As an adult, you can distinguish about 10,000 different smells. Here's how your nose does it. Smell starts when you sniff molecules from the air into your nostrils. 95% of your nasal cavity is used just to filter that air before it hits your lungs. But at the very back of your nose is a region called the olfactory epithelium, a little patch of skin that's key to everything you smell. The olfactory epithelium has a layer of olfactory receptor cells, special neurons that sense smells, like the taste buds of your nose. When odor molecules hit the back of your nose, they get stuck in a layer of mucus covering the olfactory epithelium. As they dissolve, they bind to the olfactory receptor cells, which fire and send signals through the olfactory tract up to your brain. As a side note, you can tell a lot about how good an animal's sense of smell is by the size of its olfactory epithelium. A dog's olfactory epithelium is 20 times bigger than your puny human one. But there's still a lot we don't know about this little patch of cells, too. For example, our olfactory epithelium is pigmented, and scientists don't really know why. But how do you actually tell the difference between smells? It turns out that your brain has 40 million different olfactory receptor neurons. So odor A might trigger neurons 3, 427, and 988, and odor B might trigger neurons 8, 76, and 2,496,678. All of these different combinations let you detect a staggeringly broad array of smells. Plus, your olfactory neurons are always fresh and ready for action. They're the only neuron in the body that gets replaced regularly, every 4 to 8 weeks. Once those neurons are triggered, the signal travels through a bundle called the olfactory tract to destinations all over your brain, making stops in the amygdala, the thalamus, and the neocortex. This is different from how sight and sound are processed. Each of those signals goes first to a relay center in the middle of the cerebral hemisphere, and then out to other regions of the brain. But smell, because it evolved before most of your other senses, takes a direct route to these different regions of the brain, where it can trigger your fight or flight response, help you recall memories, or make your mouth water. But even though we've all got the same physiological setup, 2 nostrils and millions of olfactory neurons, not everybody smells the same things. One of the most famous examples of this is the ability to smell so-called asparagus pea. For about a quarter of the population, urinating after eating asparagus means smelling a distinct odor. The other 75% of us don't notice. And this isn't the only case of smells differing from nose to nose. For some people, the chemical androstenone smells like vanilla. To others, it smells like sweaty urine, which is unfortunate because androstenone is commonly found in tasty things like pork. So with the sweaty urine smellers in mind, pork producers will castrate male pigs to stop them from making androstenone. The inability to smell a scent is called anosmia, and there are about 100 known examples. People with allicin anosmia can't smell garlic. Those with eugenol anosmia can't smell cloves. And some people can't smell anything at all. This kind of full anosmia can have several causes. Some people are born without a sense of smell. Others lose it after an accident or during an illness. If the olfactory epithelium gets swollen or infected, it can hamper your sense of smell, something you might have experienced when you were sick. And not being able to smell anything can mess with your other senses too. Many people who can't smell at all also can't really taste the same way the rest of us do. It turns out that how something tastes is closely related to how it smells. As you chew your food, air is pushed up your nasal passage, carrying with it the smell of your food. Those scents hit your olfactory epithelium and tell your brain a lot about what you're eating. Without the ability to smell, you lose the ability to taste anything more complicated than the 5 tastes your taste buds can detect: sweet, salty, bitter, sour, and savory. So the next time you smell exhaust fumes, salty sea air, or roast chicken, you'll know exactly how you've done it, and perhaps be a little more thankful that you can.
Right? So you can see that how we basically sense smell, right? So as it is mentioned in the text also, that we have a very densely packed olfactory epithelium, which has the extensions from these GPCRs, or G protein coupled receptors, which belong to a class of 7 helix transmembrane type of structure for receptors. Okay. Now, looking at the olfactory system, I would like your attention towards another concept, which is specifically related to GPCRs. So what are GPCRs? GPCRs are G protein coupled receptors. They have a very characteristic structure. That structure is this 7 helix transmembrane receptor. So what is 7 helix transmembrane receptors? These are the receptors which have 7 helices. Helix is a protein secondary structure which transfers the membrane 7 times. Okay. Now, this is the depicted version of the 7 helix transmembrane receptor, right? But the proteins do not look like this in real life. So to get the taste of how they actually look like, a bit at least, which we can see here, specifically for G protein coupled receptors, we can refer to this. I'm extremely sorry for the ad. I'll mute it.
G-protein coupled receptors, GPCRs, have central roles in many physiological functions and diseases.
See, you can see, right? These are the 7 transmembrane helices, and this is the membrane part of a cell.
This diverse superfamily includes over 800 members, all sharing a common configuration, passing through the membrane 7 times. GPCRs are present on all cells in the body. They pass signals from a variety of extracellular messengers to modulate intracellular pathways via the activation of G proteins and other molecules. GPCRs mediate an incredible array of essential actions in the body, including smooth motion and contraction, neurotransmission, immune regulation, and overall metabolism, and thus have enormous potential as therapeutic targets for multiple diseases. GPCRs are the largest and single most important family of drug targets in the human body. 25 to 30% of current drugs target GPCRs, including many of the best-selling drugs. Despite this, drug development has been encumbered by a lack of structural and mechanistic information about GPCRs and how compounds interact with them. The predominant problem is that GPCRs are too unstable to easily purify, and they lose their highly organized structure and activity once removed from the cell membrane, precluding them from use in structure-based drug design. To overcome this problem, Heptares developed a structure-based drug discovery (SBDD) platform to transform GPCR drug discovery. Heptares STAR stabilized receptor technology forms the backbone of its integrated SBDD platform for targeting GPCRs. STAR technology involves the selection of mutations to the protein sequence, which increase thermostability and stabilize GPCRs in their natural states. Once produced, the STAR GPCR can be purified from cells and still maintain its shape and function. The STAR GPCR can be readily crystallized together with drug hits to determine the detailed shape of the GPCR and ligand interactions, which can reveal new binding sites and opportunities. A variety of different compounds, ligands, can be screened in silico for their ability to fit the receptor. This information facilitates the precise design of drug candidates, with an ideal fit to increase selectivity and a better potential for development success. STARs can also be used to unlock the power of fragment screening for intractable GPCR targets and to help discover novel GPCR targeting antibodies, which can have advantages over oral drugs for some diseases. Through this technology, Heptares is building an exciting pipeline of new medicines to transform treatment options for Alzheimer's disease, schizophrenia, cancer, migraine, metabolic disease, and other indications.
Backtracking.
Right? So as we have seen that GPCRs are a major drug target receptors. They are widely distributed in our body, and they are involved in some of the important functions which our body performs. So targeting drugs, identifying GPCRs. So for all this function, what we need a GPCR protein, right? A protein isolated from a membrane. And as you see, you might have noticed in the video that they mentioned what is the major problem with the GPCR to understand its structure or to even understand its function. It is that it is not easily extracted from the lipid bilayer, lipid membrane. So this is true for all the membrane membrane proteins. So if a protein is of membranic nature, meaning if it is a transmembrane protein, so the moment you take the protein outside from its local environment, it will lose its structure. And the moment the protein loses its structure, it also loses its function, right? So if you might have studied about the protein structure, the primary structure of protein is what? The primary structure of a protein is basically a linear assembly of amino acids, right? So all these beads, these are amino acids, and they are just connected via direct on a single string, right? So this is the primary structure of a protein. If we talk about the secondary structure, so predominantly there are two type of secondary structures. There are many more, but predominantly there are two secondary structures which is oftenly found. First one is the alpha helix, which is a 7-helix transmembrane receptor, right? It's the alpha helix. Alpha helix means this straight chain is now like a spring. It is curved, right? So it has intramolecular hydrogen bonding, meaning within this spring-like structure, which is called a helix, it has how it come into existence as a helix or as a spring, because it has made connections, and those connections are type of hydrogen bond, right? The other secondary structure is beta sheet. So what will happen? Instead of getting coiled like this, the other combination is, if this is one chain of a protein, this is another chain of a protein, now they will interact, right? So this type of secondary structure is called beta sheet, because they'll be having multiple such proteins, right? Peptides to be specific. These will be secondary structure. Now, when these secondary structure merge with each other, then they give rise to a tertiary structure, right? And if multiple tertiary structures come together, they form the functional protein. Tertiary structure is the functional form of a protein. So the moment you separate the membrane protein outside the membrane, it loses its tertiary structure, leading to loss of function. And hence, even if we have the protein outside the membrane, its function is not characterized because it has lost its structure. So this problem is associated with all the membrane proteins, but scientists have worked hard enough, and there are multiple ways you can actually isolate membrane protein now pretty easily in comparison to previous methodology which was existent. So now, with this advent in the technology of isolating membrane protein easily and crystallizing it and understanding the structure, we can now target more drugs, right? Now, that targeting of drug, it can be either in vivo or in silico. In vivo means that in vitro or in vivo means you are doing it in a wet lab. The lab is there, you have a drug molecule, and you are testing how the cells are surviving, if the receptor is present on the cell, surviving or not surviving, right? Basic observations. Then in silico, as you have seen in the video, there are different algorithms, there are different tools which will actually tell you that if you have a structure of a protein, so computationally you can actually design small molecules, and you can see where that small molecule is interacting, and later you can confirm this observation in the wet lab experiments or through experiments on cells or in animal models, right? This is how research progresses. So now, without spending much more time on this, I guess we'll start with today's question session. Okay. So I hope that this is clear to you guys. And if not, as always, you can drop me a mail or you can put a comment in the YouTube, and I'll definitely try to respond. So for today, our first question is, and we'll follow the same scheme. I'll give you guys 1 minute for each question. Then we'll understand about the question. Then we'll see what are the options available, and if any reference is available or any reference video is available, and then we'll look into it. Okay. So our first question for today is, which of the following cell types is part of the peripheral nervous system? Is it cortical neurons, oligodendrocytes, microglia, or Schwann cells? So your 1 minute for this. Oh, okay. So your 1 minute for this particular question. Starts now. Let me put it in the chat box so that people can read. Yeah, it starts now. Put your answer in the chat box. Also, if you know the answer, good for you. You should also know about why the other options are wrong. And if you don't know the correct answer, try to eliminate the wrong answers from your memory-based, from your memory. And if you have even minimalistic information about the options. Yeah. You'll see the right answer. The right answer is Schwann cells. We are talking about peripheral nervous system and not the central nervous system. So if you do not remember what are the cell types for central nervous system and peripheral nervous system, this is a very basic classification and characterization of the cells which are present in the central nervous system and the peripheral nervous system. So in central nervous system, the most common cells, or these are the basic cells which are present in CNS, are neurons. Obviously, neurons has to be there. So what are the characteristics of neuron? Neurons are excitable. They can receive, they can process, they can transmit information. Okay, they can do all of it. What about microglia? Microglia type of cell belongs to central nervous system. And what kind of function it does? It provides innate immunity in the central nervous system. Okay. Then astrocytes. So what are astrocytes? Astrocytes maintain the blood-brain barrier integrity. They participate in synapses. Okay. Then ependymal cells. Ependymal cells are build barriers between compartments. I guess we haven't discussed much about it. So we'll keep this discussion for some later period. And if not, you guys can read by yourself about the type of cells and what are the functions oftenly associated with ependymal cells. Okay. And then oligodendrocytes, they produce myelin sheath. We have seen this in the previous week lecture itself, correct? Then talking about the cells which are present in peripheral nervous system or PNS, these are neurons. Again, same neurons, excitable. They can receive, process, and transmit information. Satellite cells, they support other cells' body and they are in supportive nature. Okay. And then Schwann cells. So what they do? They produce myelin sheath around axons. So if you remember previous week's discussion, in the CNS, the myelin sheath is produced by oligodendrocytes. In the periphery, the myelin sheath is produced by Schwann cells. What was the purpose of myelin? It was to increase the transmission rate, right? And there were node of Ranviers, then there were saltatory motion of the action potential transmission, right? So all that information is discussed in the previous week. So if you are not able to remember those things, I will recommend you to please go and watch previous week lecture. So this is not a video, but if you remember at the beginning of this session, we have seen some 3D images, right? So I am always intrigued by this. So I've also found such 3D representation of peripheral nervous system, which I would like to share with you guys. So this is it. So what it is saying the nervous system is a complex network of nerves and cells that control your body's function. It consists of the central nervous system, the brain and the spinal cord, and the peripheral nervous system, the nerves that extend from the brain and the spinal cord. So you can see that this is our nervous system, correct? Now, if you put your arrow and you select, you can actually see from different angles. You can zoom in, you can zoom out, correct? So if we talk about brain, here. So if we talk about brain, you will get a very concise information about brain. So what are the things which are associated with the nervous system? We can see that this is the brain. This is the spinal cord. This is intercostal nerve. This is median nerve. This is sciatic nerve. This is, oh, what it is. Yeah. This is femoral nerve. And this is tibial nerve, right? So these are some common nerves and major nerves which are present in peripheral nervous system. And obviously you can always read some basic details, and the moment you understand something, then you can go back to the standard books to understand the concepts and the topics in a much better way, correct? So with this, I hope that it is clear to you guys, and it was not a much of a difficult question if you know the type of cells in the CNS as well as in the PNS. Okay. So for us, the second question for today is, which of the following best describes the order of signal conduction in the stretch reflex arc? So is it sensory neurons, to spinal cord, to motor neurons, and finally to the effector? Or is it sensory neurons, spinal cord, frontal lobe, motor neurons, and then finally to the effector? Or is it motor neurons, spinal cord, frontal lobe, sensory neurons, and it lasts to the effector? Or is it sensory neurons and directly to the effector? So you guys have 1 minute for this question. Kindly drop your answer in the chat box and your time starts now. To solve such type of questions, I will recommend you guys to have again the same mind map which I was talking. And also you can put yourself in the situation if you have little bit information about stretch reflex, right? So you know the stimulus will come, then something will happen, then something will happen, right? So have that image and put yourself in the situation, put yourself in a situation where your stretch reflex is being tested, and then try to trace the path. Okay, just a suggestion how to understand these type of questions. Last 10 seconds. Yeah. So the correct answer is sensory neurons, then to spinal cord, that is interneurons, then motor neurons, the ventral horn of the transmission of the stimuli, right? And then finally the effector, correct? So if you have seen the week lecture, then you know even in the first lecture itself, the whole reflex arc was discussed. So I have just kept that image here for you guys to remember the same thing. And also for better understanding, because during the lectures, chances are if you're writing notes, then you might not be able to put them into words, because it is more inclined towards understanding the concept rather than like in the writing format. So I have tried to keep some text also in particular to today's lecture, so you guys know how what exactly the things are, and it will help you in writing subjective answers also. Okay. So the stretch reflex is accomplished through several different structures. In the muscle, there are muscle spindles whose intrafusal muscle fibers lie parallel to the muscle and sense changes in length and velocity. The afferent sensory neuron is the structure that carries the signal from the muscle to the spinal cord. Okay, it carries this action potential to the dorsal root ganglion of the spinal cord. The efferent motor neuron is the structure that carries the signal from the spinal cord back to the muscle. It carries the action potential from the ventral root of the spinal cord to the muscle down the alpha motor neuron. This synapses on the first structure discussed the extrafusal fiber of the muscle spindle, right? And we'll discuss more about intrafusal and extrafusal muscle, though we have discussed in the previous classes also. But in today's class, I guess there are 2 to 3 questions which are particularly oriented towards extrafusal and intrafusal muscle fibers or muscle spindle at the end of the day. Okay. So moving on to the next question. So what is our next question for today? The question states that muscle spindle is a blank that detects change in dash, right? So we need to fill in the blanks. So muscle spindle is a neuron that detects changes in the concentration of ions in extracellular fluid, or is it muscle spindle is a neuron that detects changes in the concentration of ions in intracellular fluid, or muscle spindle is a stretch that detects changes in muscle spindle is a stretch receptor that detects changes in length of a muscle, or last, is it muscle spindle is a stretch reflex circuit that detects the change in conduction velocity of muscle neuron. So I'll give you guys 1 minute for this question. Try to understand the question, what it is asking, and in the previous question, we have somehow discussed what muscle spindle is, so you might get some idea from it. And your time starts now. I give this 1 minute also for the people who later view this video in the YouTube, so that they don't need to pause it again and again when they are trying to attempt these questions. Ten more seconds to go. Yeah, the correct answer is muscle spindle is a stretch receptor that detects changes in length of a muscle. Okay. So what does muscle spindle mean? And we'll see short video, and I have given this reference also, which very, very nicely explained the structure, function, and the physiological importance of muscle spindle. So if you guys have time and if you're interested, you can always go back to these references, which I often mention in my slides. Yeah, so what is a muscle spindle? A muscle spindle can be defined as small, spindle-shaped sensory receptor located in skeletal muscle tissue, and they run parallel to the main muscle fibers, that is extrafusal fibers. Okay. A muscle spindle consists of several differentiated muscle fibers, that is intrafusal fibers, that are enclosed in a spindle-shaped connective tissue sac. The end of the intrafusal fiber are contractile, but the central portion is non-contractile and innervated by special neurons named gamma motor neurons. Muscle spindles are sensitive to both the phasic stretch, that is the rate at which a muscle stretches, and the tonic stretch, that is the extent to which the muscle is stretched. Stimulation of muscle spindle elicit a contraction in the stretched muscle, that is myotactic reflex or the stretch reflex, and at the same time inhibits action potentials to antagonistic muscles. The muscle spindles also participate in regulating the muscle tone at which your muscle is moving, right? muscle spindles are the one which regulate it. So this is the basic flow of a stretch receptor, that the broader ones which are more in number are the larger ones are the extrafusal one. These are the intrafusal one, which has like alpha and gamma neurons. And I'm not, because in the coming slides we have certain questions associated with it, so that is why I'm not giving you much information here. And then you can clearly see how it is being flown in the stretch reflex, right? So again, as I mentioned, we have some small video with respect to this. This second video, I guess, is the longest one, which I'm not gonna play due to time constraints. So you guys can look at this at your own, look this up according to your own convenience. So let me play this video and let me check if there are ads. I'll skip it and then I'll share it. Okay, there is an ad. I'll share the screen. It's a skippable ad. Yeah.
Proprioception is imperative in the body's ability to retain balance and posture without conscious effort. In order for the human body to orient itself in space, special sensory receptors called muscle spindles are embedded in the belly of skeletal muscle. These receptors detect changes in length and velocity of stretch. Typical muscle spindles consist of 3 types of intrafusal muscle fibers surrounded by a connective tissue sheath. These include 1 dynamic nuclear bag fiber, 1 static nuclear bag fiber, and around 5 nuclear chain fibers. As action potentials reach the muscle via alpha motor neurons, the surrounding extrafusal fibers contract or relax, stretching the muscle spindle along with them. Sensory information on the velocity of this stretch and muscle length is transmitted via afferent sensory neurons. These signals are transmitted to the central nervous system and modulated by efferent gamma motor neurons that regulate the contraction of the muscle spindle fibers. This coactivation of muscle fibers allows the body to sense and maintain its position in space. Without it, balance becomes a near impossible task.
Correct. So we can see how important it is to maintain these reflexes, because at the end of the day, we need to balance our whole body on a field, right? So if there's any issue with the sensory part of our muscle, then we might lose balance. We might get into an accident, and miserable things can happen, right? So to understand all this, it is important to know what things are associated with our system and how it works and function. That is the whole subject, right? Animal physiology. We are trying to understand how we work, basically. Correct. And if we have time in the end, then I guess I'll show this video at the end, but I guess we should move ahead and discuss all the questions. And if time permits in the end, we'll definitely come back to this video and we'll see. Okay. So our next question for today is, sorry for that. Yeah. So our next question for today is, which of the following muscle type is present in a larger number? Is it extrafusal fiber? Is it intrafusal fiber? Is it both are present in equal number, or none of the above? If you have watched the video carefully, you can answer this question very quickly. But as my protocol suggests, I'll still give you guys 1 minute for this. So do try to attempt it. Your 1 minute starts now. Last 10 seconds to go. The correct answer is extrafusal fiber. It was pretty evident, right? I have also mentioned in the last question as well as in the video, it was mentioned. These are the bigger muscles which are covering the intrafusal fibers. And in the name also, you can get some idea that intra means internal, extra means outside, right? So you can get whatever will be on the outside will be protecting something from the inside. So they have to be in the large number, right? So some little bit information about the extrafusal as well as intrafusal fiber, specifically for extrafusal fiber. So a strong stimuli applied to the body are reflected back in a muscular response, correct? These reflexes are stereotypical and hardwired. The withdrawal reflex refers to the withdrawing of a limb upon painful stimulation of the skin of the limb. The pain information is carried by the pain fibers that synapse onto interneurons within the spinal cord. These in turn make synapses on the motor neuron of both the flexors and extensors. Okay. Pain activates the flexor and inhibit the extensors. The crossed extensor reflex generally occur with the withdrawal reflex and refers to the extension of the opposite limb. Muscle have receptors for stretch and force. Extrafusal muscle fibers comprise the bulk of muscle and form the major force generating structure. Intrafusal muscle fiber are buried in the muscle and they contain afferent receptors for stretch, but they also contain contractile elements. Gamma motor neurons innervate intrafusal fiber and control their contraction. Contraction of a muscle relieves stretch on the parallel intrafusal fibers, and this defacilitates muscle contraction and also removes the effectiveness of the receptor as stretch receptor. Okay, I hope that this is pretty self-explanatory in itself. So looking at the video specifically for the structure of extrafusal fiber, because we keep talking and even in the sessions, it's a hand drawn images. So chances are you might not get the whole picture of the thing. So with that keeping in mind, I have tried.
As a tennis player, what I'm most afraid of? Buying a racket that doesn't suit your game. And I'm sure you are too. In fact,
Hello. In this video, we will see alpha-gamma co-activation, also called alpha-gamma linkage. It's an interesting topic about muscle spindle. Welcome back to nonstopneuron.com. Started. First, let's quickly revise what we have seen in the video on muscle spindle and stretch reflex. There we saw that muscle has two types of fibers. One is extrafusal fibers. They are the main force generating fibers of the muscle, and they are innervated by alpha motor neurons. The second type of fibers is called intrafusal fibers. They mainly play a sensory role as part of muscle spindle. However, their ends also contain contractile elements, so they can also contract. And they are innervated by gamma motor neurons. The central region receives sensory innervation, and it plays a major role in the stretch reflex. What happens is when a muscle is stretched suddenly, the stretch is sensed by the central region of the intrafusal fibers. Via sensory nerve, this information is sent to the spinal cord. This excites a motor neuron to the same muscle, so there will be reflex contraction of that muscle. Thus, the stretch reflex opposes a sudden change in the muscle length. Okay, that's fine. Sensory and alpha motor neurons were involved in this. But what is the role of gamma innervation in all these? Well, This innervation keeps the muscle spindle sensitive to stretch over a large range of muscle length. To understand it, first let's see what would happen without gamma innervation. Imagine we want to move our muscles voluntarily. For this, brain sends a signal via the upper motor neuron to the alpha motor neuron of the relevant muscle. It stimulates the extrafusal fibers and muscle contracts. Now the spindle is connected in parallel to the extrafusal fibers. So the contraction of muscle tends to make spindle go slack. In this state, the spindle is not sensitive to further changes in the length. And that is where gamma innervation comes in. They also receive signals from the same upper motor neuron. So normally, when alpha motor neurons are activated, the gamma motor neurons are also activated along with them. The gamma neuron causes contraction of the intrafusal fiber. This contraction helps maintain tension in the muscle spindle even when the whole muscle is contracted. And this in turn preserves the sensitivity of the muscle spindle. Thus, the simultaneous activation of alpha and gamma neurons increases the range of lengths over which spindle can work. This is called alpha-gamma coactivation or alpha-gamma linkage. This tends to make sensory endings on muscle spindle go slack and thus insensitive. But simultaneous activation of gamma motor neurons causes contraction of intrafusal fibers of the muscle spindle. This helps maintain tension and thus preserves the sensitivity of muscle spindle over a wide range of muscle lengths. That's it for this video. If you feel
Correct. So now you understand the role of extrafusal and intrafusal muscle fibers, right? So extrafusal means they are in bulk amount, they contract and relax. And then there is intrafusal fiber, which are inside the covered with extrafusal fiber. They have both sensory part as well as the non-sensory part, right? And they are innervated by different types of neuron. And this is an important information. You guys should remember this, that extrafusal muscle fibers are muscle spindle fiber are uh, uh, supplied with what kind of a neuron, and intrafusal muscle fiber are supplied with what kind of neuron. Okay. Moving to our next question. Yeah, so for today, our next question is, which of the following are sensors? Is it extrafusal fiber, intrafusal fiber, both A and B, or none of the above? You guys have 1 minute for this question, and I hope that you guys answer it correctly. And if you don't remember this, like what are sensory, which one of this is sensory, in the previous video, this was actually mentioned. So I hope that you guys are able to have a focus and concentration when you can answer this question. Last 10 seconds remaining. The correct answer is intrafusal fiber, right? So if you don't remember, go back to the lecture or go back to the previous video which I have shown you. You can easily understand why intrafusal fibers are the sensory or the sensors, right? And this was the part in the lecture video where sir has discussed the whole path as well as the role of intrafusal and extrafusal fibers. Okay. A little bit more information about intrafusal fiber, as in the previous question we have discussed about the extrafusal fiber. So a little bit information about intrafusal fiber is intrafusal muscle fibers are skeletal muscle fibers that serve as specialized sensory organs or proprioceptors or proprioceptors. They detect the amount and rate of change in length of a muscle. They constitute the muscle spindle and are innervated by both sensory, that is afferent, and motor, that is efferent fibers. Intrafusal muscle fibers are not to be confused with extrafusal muscle fiber, which contract, generating skeletal movement and are innervated by alpha motor neurons, right? So you can see the consistency whenever we are discussing about intrafusal muscle fiber or extrafusal muscle fiber, always the neurons are associated with it because this is an important information. Why these muscle fibers have this property? It is because of the neurons, right? And if you remember from the previous week discussion, or maybe from the previous week itself, we have actually seen this image itself. So you can have a brief idea, and also from the like the videos which we have just seen, we can now understand the arrangement of these muscles around a bone and how they are sensing the stimuli, how they are contracting, how they are relaxing. So if I talk about how a muscle contracts, now in this particular figure, you won't be able to justify it because here we are not talking about actin and myosin filament. We know a muscle contract when there are actin and myosin, during contraction, actin and myosin filaments are involved for muscle contraction. But how the muscle is getting the signal to get contracted or relaxed, right? It is being sent from the brain via certain neurons, which we have mentioned here, to these muscle spindle, which can sense the potential from the brain. These things will then relay their signal to the muscle, and then the muscle will do its function, whether to relax or whether to contract. So this is how the whole system is connected. So if we are understanding how a muscle contract from the point of view from a neuronal perspective, we should always keep this in mind that actin and myosin will always be associated with the muscle contraction. And for actin and myosin to be functional for muscle relaxation or contraction, we know there was a very important element which is involved. Anyone can tell me what is that element which is involved in muscle movement, relaxation, and contraction? I can give you guys options if you want. Is it magnesium, manganese, or calcium, or none of the above? If you remember. My question is, for muscle contraction, there is a very specific element which is involved for the muscle contraction. Actin and myosin function depends upon the concentration and availability of that element. So if you remember, great. If you don't worry, you can go back to the skeletal system lectures, right? So that is how we are progressing. If we are talking about neuronal system, we are understanding how a muscle contract. At the end of the day, by a muscle contraction, we are understanding how we are movement. And the moment we are talking about movement or motion, obviously the bones will be involved. And we have covered our skeletal system. What are the different types of bone, how bones are connected via ligament and tendons, and how do we move, what are the joints and all those stuff we have covered, right? So we should not see these lectures as separate lectures. We are continuing. If you want to understand a motion, for example, if I want to understand this motion of my wrist, so in this motion, I can study the motion of my wrist via skeletal system point of view because my bones are involved. From muscle point of view, because there is relaxation and contraction of my muscle, I can understand this from other perspective of what are the neurons, blood vessels, signals, small molecules are associated with it, right? So we can understand one single phenomena from different perspectives. Got it? Yeah. So next question for today is, alpha motor neurons synapse on blank and gamma motor neuron synapse on blank. So alpha motor neuron synapse on extrafusal fiber and gamma motor neuron synapse on extrafusal fiber. Alpha motor neuron synapse on extrafusal fiber and gamma motor neuron synapse on intrafusal fiber, or alpha motor neuron synapse on intrafusal fiber and gamma motor neuron synapse on extrafusal fiber. And last, alpha motor neuron synapse on intrafusal neuron and gamma motor neuron synapse on intrafusal neuron. Obviously, from this, like 2 options are wrong. So among the other 2 remaining, try to understand, try to give the correct answer. You guys have 1 minute for this particular question. You guys have 10 seconds to go, and then we'll see what is the correct answer. And I guess if you are finding it difficult to learn this, I have a very small anecdote of how I learned it basically. So it might help you guys too. The correct answer is extrafusal fiber and intrafusal fiber. If you don't remember, in the last discussion itself, we read that. Also, how I learned it, the anecdote which I was talking about. See, if you're looking from outside, what is the first thing which you're going to encounter? It will be extrafusal fiber, right? Because they are the one which are on the outside. So in alpha, beta, gamma, epsilon, theta, and all those things, alpha is the first one, right? So alpha will encounter the outermost one, that is the extrafusal one. And then we'll go inside. So alpha, beta, gamma. So this is gamma neuron. So gamma neuron will be on the inside of the extrafusal fiber, or inside is the intrafusal fiber. I know this is not the right way to learn, but this is how I made a mnemonic and I learned. So it might help you guys also in keeping this in mind. Like which type of neuron is innervating what kind of muscle fiber, correct? So some basic information about alpha motor neuron. So alpha motor neurons, also called as alpha motor neurons, are large multipolar lower motor neurons of the brain stem and spinal cord. And I hope that all these terms are clear to you guys by now, because we have discussed all this and we have covered all this about what is a multipolar, what are upper and lower motor neurons, what is a brain stem, and what is spinal cord, right? So I hope you guys are able to follow with this. They innervate extrafusal muscle fibers of skeletal muscle and are directly responsible for initiating their contraction. Alpha motor neurons are distinct from gamma motor neurons, which innervate intrafusal muscle fibers, right? Local circuitry involving sensory inputs, local circuit neurons, and alpha and gamma motor neurons are especially important in the reflexive control of muscle activity. The stretch reflex is a monosynaptic circuit with connections between sensory fibers arising from muscle spindles and the alpha motor neuron that innervate the same or synergistic muscles. Gamma motor neurons regulate the gain of the stretch reflex by adjusting the level of tension in the intrafusal muscle fibers of the muscle spindle. I guess now you guys are able to connect, right? We have just seen in a video. That is why I prefer learning via videos, because these videos are very informative. And when you read something, so if you have a visual representation of things, it becomes easier to understand when you go through the text. This mechanism sets the baseline level of activity in an alpha motor neuron and helps to regulate muscle length and tone. Okay. What happened? Okay. So this is the representation of the reflex arc, having most of the details which you guys can very easily appreciate, because you guys have learned this, you are aware of it, and I hope that you guys are even understanding it in a better way. Okay, so here is another representative video, I'll say, to understand the reflective arc and how the signal is being processed. I'll quickly share with you. Yeah.
The monosynaptic reflex arc is variously referred to as the stretch, deep tendon, or myotatic reflex, and it is the basis of the knee, ankle, jaw, biceps, or triceps response tested in a routine physical examination. These reflexive responses are too rapid for involvement of the brain and are mediated by the local circuitry within the spinal cord. The sensory signal for the stretch reflex originates in muscle spindles, the sensory receptors embedded within most muscles. The spindles comprise 8 to 10 intrafusal fibers arranged in parallel with the extrafusal fibers. The extrafusal muscle fibers make up the bulk of the muscle and are innervated by alpha motor neurons. There are two structural and functional classes of intrafusal fibers: the nuclear bag fibers and the nuclear chain fibers. These fibers differ in the position of their nuclei, the intrinsic architecture of their myofibrils, and their dynamic sensitivity to stretch. Large diameter sensory axons, group 1A and group 2 afferents, are coiled around the intrafusal fibers. These afferents are the largest axons in peripheral nerves. Because action potential conduction velocity is a direct function of axon diameter, these fibers mediate very rapid reflex adjustments when the muscle is stretched. The stretch imposed on the muscle deforms the intrafusal muscle fibers. In the afferent axon endings coiled around the spindle, mechanically gated ion channels become activated, initiating action potentials in these neurons. Group 1A afferents, which preferentially innervate nuclear bag fibers, respond phasically to small stretches, while Group 2 afferents, which innervate both fiber types, signal the level of sustained stretch by firing tonically in proportion to the degree of stretch. Small gamma motor neurons modulate the level of excitability of the muscle spindles. The sensory neurons form monosynaptic excitatory connections with the alpha motor neurons and the ventral horn of the spinal cord that innervate the same, homonymous, and synergist muscles. The sensory neurons also innervate local circuit neurons that form inhibitory connections with the alpha motor neurons of antagonistic muscles. The monosynaptic pathway from the spindle to the alpha motor neuron is unusual. Most sensory neurons from the periphery do not contact the lower motor neuron directly, but instead exert their effects through local circuit neurons. When the muscle is stretched, the afferent volley from the muscle spindle is relayed to the alpha motor neurons, and an efferent volley returns to the muscle. Since muscles are always under some degree of stretch, This reflex circuit is normally responsible for the steady level of tension in muscles called muscle tone. When the muscle shortens, the spindle is relieved of tension or unloaded, and the sensory axons that innervate the spindle might therefore be expected to stop reporting information about muscle length. However, gamma motor neurons become activated at the same time. These neurons terminate on the contractile poles of the intrafusal fibers, causing them to contract, thereby maintaining the tension on the central region of the intrafusal fibers where the sensory axons terminate. Thus, co-activation of the alpha and gamma motor neurons allows spindles to function at all muscle lengths during movements and postural adjustments.
I hope that this video was helpful to you guys so that you can understand and appreciate the role of these reflex arc and stretch reflex arc particularly, because stretch reflex arc is the one which is actually balancing us, helping us in controlling our strength. Because if you hold, for example, if you're holding a very delicate object in your hand, you do not want to put more pressure or muscle strength on that delicate object, right? On the other hand, if you're lifting weight or you are lifting something heavy, you want all of your muscle strength at the same time, right? So this all is controlled via this reflex arc, which is also controlling the tone, as we have seen, right? How much you want to put, what is the relaxation or contraction. And on the other hand, another important aspect which you should always remember, that if you're contracting a muscle, there will be other muscle which might be relaxing. So if we are talking about, for example, bicep and tricep, if I'm contracting like this, right? So maybe the bicep is getting contracted, but the triceps are getting relaxed, correct? So they always work in pair. This pair is called antagonistic pair. If you have seen in the video, right? Antagonistic is opposite to the existing one. So if something is contracting, and the antagonist of it will be relaxing, and if something is relaxing, the antagonist of it will be contracting. Okay. So I hope you guys can now have a better understanding of stretch reflex arc, because many things are repetitive, right? So I hope that you will keep this in mind, and all this exercise you're spending time here will be fruitful for you. Moving on to the next question. The next question says, which of the following is part of the inner ear? Is it ear canal, stapes, eardrum, or cochlea? I'll give you guys 1 minute as usual. Put your answers in the chat box. Last 10 seconds to go. The correct answer is cochlea. And this is the basic representative structure of the ear. And I think I might not spend much time here. I will recommend you guys to go back to the lectures if you haven't seen it. In the lectures, the structure of the ear is very well discussed, even the especially for the inner ear itself. A lot of things were discussed. So I'll recommend you guys to go back to the lectures. But here I can give you a brief and how to read this figure basically. So you can clearly see that these are color coded. So this orangish color means that it is the outer ear. This is the part which you can see to some extent, correct? Then this small pink part is the inner ear part, correct? And this blue one is the this pink one is the middle ear part, and this blue one is the inner ear part. And these are the parts of the ear. So the inner ear has a tube which is called the external auditory canal. So we basically receive sound from here, correct? So we receive sound from here. Then this is a tympanic membrane which basically vibrates, because these are kind of vibrations only. The sounds are vibrations. So this receives the vibration, it vibrates to a certain frequency. Now this is further connected to the inner ear, right? You can clearly see this oval window. By oval window, it is connected to the inner ear, and inner ear has multiple parts. That is the vestibular apparatus. You can see here the vestibulocochlear nerve, which innervates the inner ear, and the cochlea part, correct? So if you guys are not able to understand this, go back to the lecture, and even then, if the structure is not clear to you guys, I might discuss it later if you guys want it. Okay. Moving to the next question for today. Yeah. So of the vestibular hair cells in ear, which of the following is taller? Kynocelia, stereocilia, dipterocelia, or heliocelia? If you have attended the course week lecture, you will answer it. And if not, don't worry, we'll discuss. Yeah. And your 1 minute starts now. Last 10 seconds to go. The correct answer is kynocelia. And this is basically how the cochlear region, the inner region, vestibular system basically looks like. So you can see the tallest one is the kynocelia. The shorter ones are the stereocilia. Okay. So to understand the structure in a much better way and with good visualizations, we'll quickly see a small video related to the structure of the uh, inner ear, especially the hair cell. Here it is. Yeah.
The sound-sensitive cells within our ears are called hair cells. Each has a tuft of spiky extensions called stereocilia on its upper surface, and each sends signals to auditory nerve fibers through its basal surface. The hair cells are embedded in a layer of supporting cells and are sandwiched between two sheets of extracellular matrix, the tectorial membrane and the basilar membrane. Sound vibrations cause the basilar membrane to vibrate, and this motion pushes the stereocilia against the tectorial membrane. The stereocilia tilt, triggering an electrical response in the hair cell. The activated hair cell, in turn, activates the auditory nerve cells. The hair cell membrane contains mechanically gated channels. These channels are closed when the stereocilia are not tilted. However, when they tilt, a linking filament from one stereocilium to a channel on the neighboring stereocilium pulls at the channel, opening it. Positively charged ions flow into the cell, depolarizing the membrane.
Such a sophisticated way to transmit signal, right? So there is a thread, and the moment it is tilted, that thread will pull and the channel will open. It's amazing how biology works, correct? And by understanding all this, we can actually implement this knowledge in solving some of the problems of human health, human and in general living organisms. It always fascinates me. I hope that animal physiology course might do the same for you guys. Yeah, moving on to the next question. Yeah, so our next question for today is, of the vestibular hair cell in ear, which of the following are more in number? Is it kinocilia, stereocilia, dipterocilia, or heliocilia? I'll give you 1 minute for this, though this question doesn't require 1 minute, but still. Last 10 seconds. The correct answer is stereocilia, because if you remember, the tallest one will be the lower in number, right? Because there was only one representative taller one and there were multiple smaller ones, and smaller ones are the stereocilia, correct? So this is the structure. This is how they look in real under like laboratory condition where you can actually observe them. And these are the depicted, how they can be depicted. Don't worry about these channels. This is some extra information because you guys are aware of channels, transporters, especially calcium, potassium, right? You guys are also aware of potential, action potential. You guys are also aware of afferent and efferent axons, right? So just for connecting all the information, I have kept this figure, but this figure is mostly for understanding the kinocilium, that they are lower in number and there are multiple stereo cilias. Okay. So let me see if this video is relevant for now. If we have already covered the topic, I won't show this. You guys can see this in your free time when you guys are going through these lectures. Okay, yeah, it was not directly relevant. You guys can see this later, I guess. It is mostly related to deafness. What are the causes of deafness and how we can overcome some kind of deafness. So you guys can go and check this video out later. Okay, yeah. So for today, our last question is, otolith present in ear is made up of hydroxyapatite, potassium oxalate, sodium chloride, or calcium carbonate. You guys have, as usual, 1 minute to answer this question. And I hope that you guys at least try to attempt it. Your 1 minute starts now. Last 10 seconds. The correct answer is calcium carbonate. So if you guys don't remember or you haven't gone through the lecture videos, again, same thing. Please watch the video lectures and then attend the live sessions. They will be more fruitful to you guys. Yeah. So an otolith, also called statoconium or otoconium or statolith, is a calcium carbonate structure in the sacculae or utricle of the inner ear, specifically in the vestibular system of vertebrates. The sacculae and utricle in turn together make the otolith organs. These organs are what allows an organism, including humans, to perceive linear acceleration, both horizontally and vertically. They have been identified in both extinct and extant vertebrates, right? So if you are moving in a car, you feel nauseated. If you're going in a lift up and down, there's sudden change, right? You are perceiving it. That is because of the otolith organ in your inner ear, which basically senses all this information. So we will see this video because it is relevant to the current topic. Yeah, let me skip this.
The vestibular system is responsible for the body's equilibrium. It maintains balance and provides awareness of the body's spatial orientation. Vestibular sensory organs detect changes in the head's positions and movements and transmit this information to various regions of the brain. Projections to the brainstem trigger reflex pathways that lead to compensatory actions to maintain stability or reestablish equilibrium, while projections to the cortex provide perception of gravity and movement. Examples of vestibular reflex pathways include the vestibulo-ocular reflex that controls eye muscles to keep visual objects in focus while the head is moving. It does so by moving the eyes in the opposite direction as the head. And the vestibulo-spinal reflex that senses a potential loss of balance and activates body muscles to keep the body from falling. The sensory part of the vestibular system is located in the inner ear on each side of the body. It consists of 3 semicircular canals that sense rotational movements, such as when the head is turning, and 2 otolithic organs that sense head positions, as well as straight-line motions, such as when riding in a car or elevator. The 3 semicircular canals, or ducts, are oriented approximately at a right angle to each other, each corresponding roughly to one of the 3 planes of motion, turning left and right, nodding up and down, and tilting to a side. They contain a fluid called endolymph. Each canal has an enlargement at one end called an ampulla. Within the ampulla, there are hair cells embedded in a gel-like structure named cupula. When the head turns, the ducts that are located on the same plane of motion rotate, but the fluid lags behind because of inertia. This causes the fluid to briefly move in the opposite direction as the head and either push or pull on the cupula, bending the cilia on the hair cells and thus activating them to send nerve impulses to the brain. The direction of the bend determines if the signals generated are excitatory or inhibitory. Because the 2 sides of the head are mirror images, a head turn generates excitatory signals on one side and inhibitory signals on the other. The 2 otolithic organs are 2 patches of hair cells oriented nearly perpendicular to each other, the saccule being vertical and the utricle being horizontal. The cilia of these cells are embedded in a gel-like layer sprinkled with calcium carbonate crystals called otoconia, commonly known as ear rocks. The crystals add weight to the layer, pulling it down with gravity. When the head is in upright position, the gelatinous layer bears down evenly on the cells of the utricle. The cilia remain straight and no signals are generated. On the vertical saccule, however, the heavy gel is pulled down by gravity at one end, bending the cilia, generating nerve impulses. The reverse is true when the head is horizontal. Other head positions are determined by a combination of signals coming from both organs. Vestibular sensory organs detect not the motion itself, but changes in the rate of motion, specifically acceleration or deceleration. For example, when a person is sitting in a car that starts to move, the heavy gel-like layer of the utricle lags behind at first, bending the cilia back, activating the hair cells. The more sudden the car starts, the greater the stimulation. Once the car is in stable motion, the gel catches up with the rest of the tissue and no activation results. Similar events occur in the saccule during an elevator ride, up or down.
Correct. I hope that it was fascinating for you guys too. This is how we are basically sensing our position or spatial coordinates, where we are, how we are we sitting straight or tilted, and all this everything is happening in the otolith, or the inner part of the ear. Correct. So I hope that this session was as exciting for you guys as it was for me. Thank you so much for your patience, and we'll meet next Saturday same time. Before going, as you know, I always have a question for you guys to think upon. So for this particular week, I have this question for you. So in some situations, sometimes we act in manners that we later realize if we would have acted slow or would have thought before acting, our actions would have been less aggressive or completely different. Are these the situation where our system cannot decide whether to use reflex system or central nervous system to act? Right? I hope you got the question. The question is basically saying when we are in the decisive mode, there are certain times when we might have ran too fast or ran too slow, right? Especially while playing sports. If you're playing badminton, there are certain times when you need to move really fast. And sometimes you need to just move. And there are certain acts, for example, cycling, swimming, all these things. I'm just giving you guys hint. There are certain actions which are core in CNS, and there are certain actions which can be worked via reflex system. So just think about it, and I hope so that you guys get a good answer for this question. That is all from my side, especially for today. Thank you so much. Thank you for your patience and for giving me your time. Thank you.